Hello and welcome to Self-Assembled Polymeric Nanoparticles for Drug Targeting to Tumors. I'm Shel Ip, Field Application Scientist at Precision Nanosystems. It's my pleasure to introduce today's speaker. Dr. Shyh-Dar Li is an Associate Professor at the Faculty of Pharmaceutical Sciences, University of British Columbia. He holds the Angiotech Professorship in Drug Delivery. His research currently focuses on engineering nanoparticles for drug targeting to tumors. Before we begin, some basic housekeeping. If you have technical issues, please use the question or chat box and we'll do our best to troubleshoot. If you have any questions for our speaker, please use those same features and we'll try to answer as many questions as possible at the end of the session. If we do not get to your question, we'll answer them after the event by email. We would also like to make you aware of an upcoming event we are hosting in Seattle on July 16th. Please join us at the Washington Athletic Club for the Nanomedicine Symposium, Materials, Manufacturing, and Therapeutic Applications. We will have a number of industry and academic speakers participating. We will also be demoing our entire product suite, including the NanoAssembler SPARC, a new low-volume instrument designed to manufacture nanoparticles at a microliter scale. The NanoAssembler benchtop instrument, our current instrument for rapid nanoparticle development at the milliliter scale. Also the NanoAssembler Blaze, a new preclinical scale-up instrument for batches of 10 milliliters up to 1,000 milliliters, with the option of automated dilution post-manufacture. Finally, we'll also showcase the technology behind our continuous flow GMP scale-up system that uses 8 microfluidic chips in parallel to manufacture 25 liters in 4 hours. Registration is now open. Please visit our website for more information. Now, without any further delay, Dr. Li, take it away.
Okay, thank you so much, and thank you everyone for attending this webinar, and thank you for PNI for providing this opportunity to for me to speak about my work. So today I'm just going to introduce one of the major projects in my lab. That is a polymeric micelle-based delivery system for drug delivery to tumors, and we specifically focus on water-insoluble drugs. And these drugs are usually very cytotoxic, very toxic, and so it requires specific delivery to tumors to enhance the efficacy and, in the meantime, reduce the toxicity. So the first slide just summarizes our design, our rational design for this delivery system. So we work with polymer-based systems, and so the polymer system we work with right now is a bioeliminable and biocompatible but non-biodegradable system. So we hypothesized that with this system, we should be able to improve the vehicle stability because these polymers are not biodegradable, so they can maintain their integrity during the blood circulation, so they can have a higher chance to reach tumor to do sustained release of the drug. And so another approach we use is we chemically conjugate these water-insoluble drugs into the polymers. So the reason we use chemical conjugation is that we want to produce sustained drug release rather than using a passive isolation method that usually contributes to a very rapid drug release. So that could, with this mode of delivery, we hope to increase the improve the pharmacokinetics. And so with that, we really need multiple conjugation sites in the polymer so that we can conjugate a lot of drugs per polymer. So, so we can improve the drug loading efficiency. But in the meantime, we also increase or I should say reduce the use of the vehicles. So we can reduce the cost. In the meantime, also reduce the possible potential side effects from the polymer. Okay, so the polymer system we work with is carboxymethyl cellulose. So this is also so-called CMC. So this is a very commonly used pharmaceutical excipient. So it's basically a polysaccharide polymer, but the linkage between each or the linkage between the sugar molecules is beta linkage, so it's not biodegradable. So it should have improved stability in the blood circulation. And then we did a lot of chemistry to make our own delivery system. So the first model drug we work with is docetaxel. So you can see the chemical structure down here is a very bulky chemical compound. So it has very little water solubility and requires drug delivery. So again, we did some chemistry just to conjugate this drug into our polymer system. So the starting material is sodium salt of CMC. So the first chemistry we did was to convert the sodium salt into the free acid form. And then with that, we can acetylate all the hydroxyl groups in the CMC. And so the acetylated CMC then becomes a solvent soluble and is no longer gelling. So then we can use this material to conjugate with PEG, a soluble polymer, and docetaxel, an insoluble anti-cancer drug. So in here, this is a one-part reaction. This is called EDC coupling reaction. So very simple amine esterification chemistry here. So so basically, we just mix acetylated CMC with PEG and docetaxel in one part with very little amount of water here, so that PEG and docetaxel can chemically conjugate it to the acetylated CMC. So I want to emphasize here, if we get the balance right, which is the drug, hydrophobic drug, to the hydrophilic polymer PEG right, then these the polymer will self-assemble into nanoparticles. So we actually did a lot of optimization, and we found out the optimal composition. With our NMR characterization, it shows that the optimal composition is actually contains 35% weight docetaxel and 5% weight PEG. And our UV analysis also confirms the composition of docetaxel. And our GPC analysis confirms that with our chemistry and the whole fabrication process, we can remove free or unreacted materials, including docetaxel and PEG. So this just shows the TEM structure of a Cellax nanoparticle. The final optimal composition, we call it a Cellax. So the Cellax nanoparticles is about 120 nanometer in size. But I just want to emphasize that this self-assembly process has to be well controlled in order to produce reproducible nanoparticles with good quality. That's how we use NanoAssembler to prepare our nanoparticles in a very controlled manner, so that we can ensure that each batch is in good quality for our efficacy studies or pharmacokinetic studies. So the first study we did was actually to compare the pharmacokinetics with our system, Cellax, and other taxane formulations used clinically, including Taxotere. So in the Taxotere formulation, they have to include detergent and an organic solvent, ethanol, to dissolve the drug. So with that, the detergent Twin 80 is actually causing some hypersensitivity reactions. So this formulation is not that friendly. So patients have to be pre-medicated with steroid just to reduce the hypersensitivity reactions. And there's another taxane formulation called Abraxane. This is a albumin paclitaxel complex. So the delivery mechanism is passive association. So the drug release is very fast. The advantage, the major advantage of Abraxane is that it removes or eliminates the need of incorporating a detergent in the formulation to improve the solubility. So, so the hypersensitivity reactions caused by Abraxane is pretty low. So with that, patients can receive a higher dose without significant toxicity. So with that, they can actually increase the efficacy. So the first study we did, again, is the pharmacokinetics. So you can see that both Taxotere and Abraxane formulations got cleared from the blood circulation very quickly. You see a very rapid elimination. And so this is about 3 days. We can no longer detect the drug concentration in the blood. And but if you look at Cellax, it circulates much longer than the other two clinically approved formulations. And even, I believe this 7 days after post-injection, we still have quite a bit of Cellax circulating in the blood circulation. And we can also maintain a quite consistent free drug level in the blood. This again confirms that the Cellax formulation is long circulating and a sustained release formulation. So in the right-hand side, you can see the pharmacokinetic table. So this is just summary, and you can see that the T half-life is prolonged by more than 5-fold, and the AUC is increased by about 40-fold compared to the other formulations. So again, this just shows you that our Cellax formulation is able to prolong the pharmacokinetics and also perform sustained release. So we are very interested in looking at whether this delivery system is able to improve the tumor delivery. So again, we compared Cellax delivery in the tumor with Taxotere and Abraxane. And this figure just indicate that Cellax improves the delivery, the tumor delivery by 5 to 10-fold compared to Abraxane and Taxotere. So, so we were encouraged by the data, and then we move on to test the efficacy of Cellax against a bunch of different tumor systems or tumor models in mice. So I'm just going to show you two preclinical models we tested. But we actually tested a lot of different animal models. So the first one is melanoma model in subcute. So this is a very aggressive model. But basically we can see that Cellax is more efficacious compared to Abraxane than Taxotere. And so in here you can see that Cellax flattened the tumor for for about 10 days. This is just one single injection, so it's very encouraging data. And so we also tested Cellax in more advanced tumor models, and this is MDA-MB231. Human breast orthotopic model. So these tumor cells are inoculated in the mammary fat pad in mice, and over time we let them grow, and then they will also metastasize to the lung, and also many other tissues, but mainly in the lung. So in here, this is H&E study staining on the lung tissue. So I believe this is 100 days after inoculation. So very long process for these tumors to metastasize. And so you can clearly see that with controlled treatment, docetaxel or Abraxane treatment, we still have lots of lung nodules in the lung. But with Cellax treatment, it's much cleaner. And we also use the definient image analysis tool just to quantify the metastasis in the lung. And so this imaging tool allows us to rapidly and very sensitively identify those nodules and quantify them. So in the C panel, you can see the quantification data, and just showing that with Cellax treatment, we can only detect very little amount of breast cancer nodules in the lung. But compared to docetaxel and Abraxane, these two treatments actually did not significantly reduce the lung metastasis. So again, very positive results. And we later on found out that the anti-metastasis effect of Cellax is actually related to its delivery mechanism. So I will talk about that in just a little bit, but first I want to introduce another piece of data. So this is done in a patient-derived pancreatic tumor model. So we know that in the tumor microenvironment, it's not just tumor epithelial cells, but also many other cell populations in the tumor. So in here, we can actually classify tumor microenvironments into 4 major cell populations and identified by different cell markers. So the first one is endothelial cells stained with CD31. The second is the cancer-associated fibroblast stained with alpha smooth muscle actin, also called alpha SMA. And the third population is macrophages, or tumor-associated macrophages, stained with F480. And the last one is the tumor epithelial cells, and stained with cytokeratin. And in this study, we actually labeled Cellax with a red fluorescent tag. And we stain different cell markers with green fluorescent agent. And the blue is the nuclei staining with DAPI. So in here, you can pretty much appreciate that these Cellax nanoparticles are mainly associated with alpha SMA positive cancer-associated fibroblasts and F480 macrophages. They have little interaction with endothelial cells and epithelial cells. So this is quite interesting. So then we later on want to understand how this is impacting the therapeutic effect of Cellax. But first, this is a little background about tumor microenvironment, or also we call it tumor stroma collectively for those normal cells recruited to the tumor microenvironment relative to the tumor epithelial cells. So again, in the tumor, it's not just tumor epithelial cells, but many other normal cells. But tumor cells actually secrete a lot of growth factors and cytokines to recruit, in the meantime also activate them. So upon activation, they actually proliferate and differentiate, and also they gain new functions. So for example, endothelial cells will grow new blood vessels to promote the tumor growth and metastasis. And for inflammatory cells, mainly macrophages, when they are recruited, they differentiate into the M2 type to promote the inflammation in the tumor. But in the meantime, it's actually promoting angiogenesis and a lot of tumor progression. And also, I want to emphasize fibroblasts. So fibroblasts here, when activated by tumor cells, they can secrete a lot of growth factors and cytokines to promote tumor migration and metastasis and progression. But in the meantime, they also produce lots of ECM, extracellular matrix, to form a niche for tumor epithelial cells to grow and progress. and they also secrete lots of cytokines, as I just mentioned earlier. So these are all very important cell populations in the tumor microenvironment. So what's the impact of Cellax affecting or killing macrophages and fibroblasts? Okay, so this is a summary about the previous slide. So tumor stroma. In this specific aspect, I emphasize on the fibroblasts, cancer-associated fibroblasts. Again, they promote tumor progression. They produce ECM, and because they proliferate a lot, they also increase the interstitial fluid pressure. And this is a very important feature in the tumor because it reduces blood perfusion and also reduces drug penetration. So it really has a significant negative impact on the tumor drug delivery. So this seems like fibroblast seems like a very important therapeutic target for cancer therapy, and we show that Cellax was able to target alpha SMA positive stroma in the pancreatic tumor, but does it kill fibroblasts in the tumor? So this is a study we did. So again, we compare Cellax activity against Abraxane in reducing alpha SMA population in the tumor. So we also counterstand CD31 as a negative control. In here, we see that there's no significant effect from Abraxane or Cellax in reducing the blood vessel density in the pancreatic tumor microenvironment. But for alpha SMA, you can see that Cellax has much higher activity against cancer-associated fibroblasts. Abraxane also has some effect, but not as profound as Cellax. So in the right-hand side, you can see the quantitative results. So this just confirms the imaging results that Cellax was more efficacious in reducing the alpha SMA positive population in the tumor, and is significantly more profound in one model compared to the other. So in both models, Cellax shows enhanced activity against cancer-associated fibroblasts. Okay, so as I just talked about, fibroblast is very important in controlling the tumor perfusion. So if the fibroblast population is depleted, then we will expect better tumor perfusion. So in the left-hand side, we show the results that Cellax was able to improve the tumor perfusion. So fixed lectin was a marker to stem perfused endothelial cells. So in the meantime, you can also see that these blood vessels are more open after Cellax treatment. Again, confirming that once the cancer-associated fibroblasts are removed, the blood vessels will have less pressure, compression pressure, and so they will be able to open and allow blood vessel to go through the tumor. And this should be able to show improved drug delivery. Our next plan will be to study once after fibroblast depletion by Cellax, can we improve delivery of another anti-tumor agent to have a synergistic effect. Okay, so on the right-hand side, we actually did another analysis just to look at the metastasis. So as we have just discussed, fibroblasts are very important for to help tumor to metastasize. So we actually look at the metastasis of the pancreatic cancer after different treatments. So with saline treatment, you can see lots of metastasis to the peritoneal cavity. And so it represents in the blue bars. And Abraxane was able to reduce the metastasis a little bit, but not as good as Cellax. So for example, with Cellax treatment, we were able to completely remove the diaphragm metastasis, and this is very important because this affects the breathing of the mice, and this will be able to improve the survival of the mice. And we also show reduced hepatic portal metastasis, and this is usually one of the major metastasis sites for pancreatic cancer, and are often affected by this kind of metastasis. So the results are very positive. And then we were wondering why Cellax are more selective to αSMA positive fibroblasts compared to other cell populations in the tumor. So we did a lot of mechanistic study just to get this preliminary data shown in this slide. So as I showed earlier, that Cellax was able to prolong the blood circulation of the drug. But in the meantime, we also found that Cellax has very high affinity with serum albumin. And serum albumin is a very interesting molecule because it recognizes multiple cell markers. And one of them is called SPARC, stands for secreted protein acidic and rich in cysteine. So this molecule is actually very important for tumor remodeling or tissue remodeling in general. And this molecule is found highly expressed in the tumor stroma cells, mostly in fibroblasts. And we also found that albumin and SPARC are critical or required for internalization of Cellax. So this is our hypothesis so far. So during the blood circulation, Cellax nanoparticles absorb a lot of serum proteins onto the surface. And upon extravasation into the tumor tissue, these albumin-bound Cellax nanoparticles were recognized by SPARC, secreted by αSMA positive fibroblasts. And somehow SPARC would be able to trigger endocytosis by the fibroblasts and bring the whole system into fibroblasts. And within the fibroblasts, docetaxel is released to kill fibroblasts. So this is so far our hypothesis. And we actually got one piece of data to support this hypothesis. And this is done in an in vivo study. So we actually compare Cellax efficacy in different models, in different tumor models. And these tumor models express different levels of SPARC. And you can see that the tumor efficacy or Cellax efficacy against tumor is positively correlated with the SPARC level in these tumors. So again, suggests that SPARC is an important molecule mediating Cellax activity. And maybe this can be used in future clinical trials, that SPARC might be a predictive biomarker that we can use to stratify patients that would likely respond to Cellax treatment, so that we can increase the likelihood of success in clinical trials. So that's the story of our first nanoparticle, Cellax. So again, Cellax is delivering docetaxel. It shows quite a bit of advantages compared to the clinically approved taxane formulations. But we also found that there are several drawbacks of Cellax, and we think we should further work on to improve Cellax. First one is Cellax shows a significant liver uptake. So in this figure, you can see that these red fluorescently labeled Cellax accumulate a lot in the liver, in addition to tumor. And so this causes some concerns of safety, because we know that a lot of uptake is triggered by macrophages in the tumor, also so-called Kupffer cells. But we are not concerned about the toxicity to the Kupffer cells because they regenerate quite efficiently. But the problem is once macrophages digest Cellax nanoparticles, it can release a high concentration of docetaxel and affecting the neighboring hepatocytes. And indeed, we saw some hepatocyte toxicity, liver inflammation, mild liver inflammation caused by Cellax. And this kind of toxicity is not favored because it could cause irreversible toxicity. So this is one of the major concerns for Cellax. And second, as you can see here in the bottom right-hand side figure that shows the tumor uptake of Cellax nanoparticles, they mainly localize in certain parts of the tumor. So this suggests that the tumor penetration of Cellax is not very significant. They always localize close to the blood vessels. And so this creates concerns about complete elimination of tumors. And also, we have tested Cellax against a number of resistant tumors, and they show inconsistent activity. So sometimes it works, but sometimes it doesn't. But we know that a delivery system or a new drug would be most valuable if it can treat resistant or refractory tumors. So we do have three aspects we want to improve for this first generation of Cellax. So our approach is we want to deliver a different drug. And we hope to work with, or we plan to work with a cytotoxic drug that is not sensitive to resistant mechanisms in the tumor, especially P-glycoprotein that is found responsible for multi-drug resistance in many tumor samples, clinical tumor samples. So in here, we actually compare the activity, or IC50, of different anti-tubulin drugs against various MDR cell lines. So we have murine breast tumor cells, we have human prostate tumor cells, and we have human breast tumor cells. And they all express either P-glycoprotein or multi-drug resistance associated protein, or beta-3 tubulin. These are identified as the major MDR markers for taxanes and antitubulins. So including docetaxel, paclitaxel, and vincristine. But in our IC50 test, we show that Podophyllotoxin remains active against these highly resistant tumor lines compared to the other antitubulin drugs. And the IC50 is in the low nanomolar. And the chemical structure here shows that it's very simple, and it only contains one hydroxyl group. And so this makes Podophyllotoxin an ideal candidate for our system because we only have one hydroxyl group, so the chemistry, the EDC coupling chemistry would be much cleaner compared to other compounds that contain multiple hydroxyl groups. So again, we use the same system to deliver Podophyllotoxin. And so with this case, we actually did a lot of optimization. So the major optimization we did was to study different drug to pack ratio. As I talked about, nanoparticle self-assembly is driven by the balance between the hydrophobicity and hydrophilicity of the whole polymer. So the ratio is very important to control the nanoparticle's self-assembly. So in the left-hand side figure, we found out that the drug-to-pack ratio controls or determines the final particle size. So when we have very low drug-to-pack ratio, meaning lower drug content but high pack content, then we got particles with smaller size, about 20 nanometer. But as we increase the drug content compared to pack content, we can see significant increase in size. And in this case, if we increase Podophyllotoxin to pack ratio to 20 more to more ratio, then we see that the particle size can be increased to 120 nanometer. And so the right-hand side just shows the 20 nanometer composition. So again, they are very homogeneous if we manufacture them using nanoassembler. So this slide shows tissue distribution depends on the nanoparticle composition. So again, we have 20 nanometer that contains low drug-to-pack ratio, all the way to 120 nanometer that has a high drug-to-lipid ratio, high drug-to-pack ratio. And we label these nanoparticles with a red fluorescent dye, and then inject them into tumor-bearing mice. So this time scale doesn't show it well, but this is from the early time point all the way to the late time point, 96 hours. So we mainly examine 3 tissues because they are the major uptake tissues for these nanoparticles. So including tumor, liver, and spleen. So you can appreciate that the 20 nanometer particles actually do not go to the macrophage tissues, including liver and the spleen. They mainly accumulate in the tumor from very early time point to 96 hours. On the other hand, if the particle size is bigger, containing more drugs, less packed, then the particle is less shielded, and so they are recognized by the macrophages in the liver and spleen, and you can see very high uptake by these 2 tissues compared to the tumor. And the 30 nanometer nanoparticles, which contain intermediate drug-to-pack ratio, actually sit in between. And in the right-hand side, it just shows the quantitative results. But I just want you to focus on the D panel that is showing a tumor-to-liver ratio at different time points for different size of nanoparticles. And you can appreciate that the 120 nanometer actually shows very low ratio, meaning very high liver uptake but little tumor uptake. But when we use 20 nanometer nanoparticles, you can see the ratio is way above 1, showing higher tumor uptake compared to liver uptake, and is that case from very early time point all the way to 96 hours. So very encouraging data for us, because liver uptake is always an issue for various types of nanomedicines. And it seems like with our system, we were able to reduce the tumor uptake significantly. So another drawback of Cellax is that it only accumulates around the blood vessel, doesn't travel far. This creates a significant problem or concern for nanoparticle therapeutics. So in this case, we actually look at how size is affecting the tumor penetration. So in here, again, this is not showing well. The top one is 20 nanometer, the middle one is 30 nanometer nanoparticles, and the bottom one is 120 nanometer. And so these tumors are collected 48 hours after injection, and these nanoparticles are labeled with red fluorescent dye. And we also, in the meantime, we also stain the blood vessel green, so we can differentiate the hypovascular core from the hypervascular rim. And so in here, you can see that 20 nanometer particles actually prefers going to the core, the hypovascular core, relative to the hypervascular rim. Compared to the 120 nanometer particles, you don't see much in the core, but relatively you can see a higher amount in the rim around the blood vessel, the green blood vessel. So maybe on the right-hand side is more clear when we show the quantitative results. So the B panel is that the overall tumor uptake, again, this just confirms the previous slide, the previous results, that 20 nanometer is much more efficient in delivering into tumor compared to 30 and 120 nanometer. And the C panel just shows that for the 20 nanometer particles, they actually preferred going to the core rather than the rim. And in fact, 90% of these 20 nanometer particles prefer staying in the core. But when you look at 30 nanometer and 120 nanometers particles, they prefer staying in the rim, so they don't penetrate that much. So again, this new system seems like having better tumor penetration if we can reduce the size to very small, about 20 nanometer. And with these tumor uptake data, we later on proceed to do the efficacy study. So this is a sub-Q tumor model, EMT6AR1, very resistant, highly expressed of P-glycoprotein. And so we compared different treatments, including the polymer control, docetaxel, free Podophyllotoxin, cabazitaxel, which is another taxane drug that is less sensitive to P-glycoprotein mediated MDR. And we also compared 3 different constructions of Podophyllotoxin nanoparticles from 20 nanometer, 30 nanometer to 120 nanometer. So in this figure, you can clearly see that the 20 nanometer Podophyllotoxin nanoparticle is the most efficacious treatment compared to the others. So we can cure 7 out of 10 mice with this very aggressive tumor model. The other formulations are not as efficacious as this 20 nanometer formulation. We can see that 120 nanometer formulation is only marginally better compared to the free Podophyllotoxin. And the clinically approved treatment, including docetaxel and cabazitaxel, are not showing significant efficacy in this tumor model. So again, very encouraging data. And we also look at the pharmacokinetics. We measure the drug. We not just image the tumor, but we also measure the drug concentration in the blood. And so again, the free drug gets eliminated very quickly, but when formulated with this 20 nanometer nanoparticles, now we call Celludo, then the pharmacokinetic is much prolonged. So as indicated by the T half-life, we are able to improve or increase the half-life from 1 hour to 24 hours, so very long circulating. And the AUC with the Celludo treatment is increased by 300-fold. So with this, we believe the efficacy improvement is anticipated. And the next model we tested is tumor metastasis. So we all know that patients are killed by metastasis, and this tumor model is established by IV injecting EMT6AR1 into mice, and these tumor cells localize in the lung and forming metastasis. So in a proof of concept study, we actually use xenogene imaging just to confirm these nanoparticles or Celludo nanoparticles accumulate in the lung metastasis. So we have three different groups. So A group is mice containing lung metastasis, but didn't receive di Celludo. So di is the red fluorescent dye. And B is normal mice without any metastasis in lung, but they received di Celludo. C is mice with lung metastasis but received di Celludo. And you can see that these di Celludos accumulate only in the C group, showing that they selectively accumulate in the lung nodules. And we have imaging data confirm the result, but I will have better results in the next slide. But first, I just want to show you the efficacy, which is indicated by the survival of mice. So without treatment, or with the standard treatment, including docetaxel or the free Podophyllotoxin, the animals did not survive long. So this within 10 days after inoculation, they all died. So very aggressive model. But with Celludo, they survive by 20 days. Some of them survive by 30 days. So very significant improvement in survival. So in here, I also want to show you the evidence that diacellulose are very selective for lung tumor nodules. So the left-hand side panel is the normal lung with diacellulo treatment. So we use Ki67 as a marker for tumor cells or for proliferating cells. So most normal lungs do not proliferate, so they are negative for Ki67 staining. And we inject diacellulo into these normal mice, and we don't see any accumulation of diacellulo in the lung. But in the right-hand side, when these mice do have lung metastasis, you can see very significant overlay of diacellulo with the Ki67 proliferating cells, just suggesting these diacellulos are very selective for lung metastasis. So the next model is a very difficult to treat model. So this is MDR, ovarian cancer model in the peritoneal cavity. So again, our data shows that diaceludo accumulates very efficiently in the ACI-ADR-RES ovarian model, and very significant uptake, but very little uptake by the liver. And with that, we can prolong the survival of mice bearing intraperitoneal metastasis of NCI-ADR-RES. So as you can see, the control polymer control treatment for the median survival of these mice is 47 days, and the second treatment group is pegylated liposomal doxorubicin, which is the standard therapy for refractory ovarian cancer in human patients. It doesn't improve the survival at all. And the same for Podophyllotoxin and paclitaxel. They do not improve the survival. But Celludo is able to prolong the survival from 50 days to 70 days. And this is a very significant improvement. So summary, Cellax displays 8-fold prolonged pharmacokinetics and 10-fold increased tumor delivery compared to Abraxane. And Cellax demonstrates improved efficacy in multiple tumor models compared to Taxotere and Abraxane. And Cellax targets tumor stroma, modulates tumor microenvironment, and reduces tumor metastasis. But the first generation Cellax is actually delivering docetaxel. And we found three major drawbacks, including high liver uptake, poor tumor penetration, and inconsistent efficacy against multi-drug resistant tumor. So we developed the second generation of drug is called Celludo. So this Celludo is much smaller size, and it delivers a drug that is active against multi-drug resistant tumors. And so we use the same system and create Celludo. And this system shows prolonged pharmacokinetics, improved tumor selectivity, and enhanced tumor penetration. And Celludo is effective targeting tumor metastasis and prolonging survival of animals. And I want to emphasize that Celludo is very active against multi-drug resistant tumor models. Okay, so that's about my presentation. I just want to acknowledge my lab. A lot of talented people. And this project is actually initiated by a former postdoc, Mark Ernstein, and other postdoc members in my lab. And right now, I have one postdoc and one graduate student working on this project. And also, I want to thank my collaborators in Toronto, and also my funding from UBC, OSCR, and CHR, and NIH. And thank you very much for your attention, and I will be happy to take any questions.
Thank you, Dr. Li, for such an engaging talk and some rather profound results. We do have time for a couple of questions, and I'm going to pick a few here for you. The first one is, I guess, about the Celludo particles. Are there any other factors apart from the PPT PEG ratio that influence the size? And I guess this is referring to conditions of formation of the particles.
Yes, we actually use the nanoassembler device to produce different kinds of nanoparticles, even with the same composition. So really, a lot of factors can control or can result in different particle size. For example, if we use different solvent to water ratio. So remember, the nanoparticles formation is actually caused by a rapid mixing of the organic phase containing the polymer and the aqueous phase. So the ratio of the organic phase and water phase is very important. So another thing, another factor that is very important is the speed of mixing. So we prefer very high or very rapid speed to mix these two these organic solvent phase and the water phase, so that we can have more defined nanoparticles with smaller polydispersity index. So these are other two factors we really need to control to get good quality particles, in addition to the drug to pack ratio.
Thank you. And the next question is about targeting. Have you ever tried any targeting ligands in order to direct the biodistribution of your particles?
We haven't tried different targeting ligands to help the targeting of the nanoparticles so far. So there are multiple factors to consider. The first one is the chemistry. Will it be compatible with our one-part reaction chemistry here? Right. So if we consider antibody, so maybe this antibody conjugation can be only done post the formation of the nanoparticles, because antibodies are very sensitive to solvent. And we also need to consider the solubility of the ligand, so that whether it would fit with our system or it would affect the self-assembly process. So that is the first aspect we need to consider. That is chemistry. The second part is can ligand affect the penetration or the targeting of the tumor? So it's still controversial, because some people said that the ligand doesn't actually help targeting or change the biodistribution that much compared to the normal or non-targeted nanoparticles, because it's still driven by the enhanced permeability and retention effect of the nanoparticles. So the ligand effect only comes after astrobization of the nanoparticles in the tumor microenvironment. But in that process, it actually limits the penetration of nanoparticles, because once these nanoparticles astrobizate into the tissue, meaning they leak out from the blood vessel and into the tumor tissue, they tend to bind with the first line of the cells they encounter. So with that, they actually don't travel that far from the blood vessel. So that's another thing we need to be careful about. And maybe there's some balance between how we can target the tumor cells better, and how we still maintain the good tumor penetration. So I think that's still a very challenging topic in this field, and we are planning to do some research on this topic.
Thank you for that insight, Dr. Li, and that's certainly true for solid tumors, and maybe access to ligands on the surface of circulating tumors might be slightly different. Yeah. Okay, thank you all for joining us for this webinar today. That's all the time we have for questions. If we did not get to your questions, I apologize, and we will get to them by email after the event.